Okay, this is a brief presentation on hereditary colon cancer. This is only a couple slides because what we're going to do is just talk about inherited colon cancers. I know that pathology has gone into a lot more detail, but in general, the risk of colon cancer is about 5 to 6%, and it's typically a slow-growing cancer that starts as a precancerous polyp. I always say that, the difference between getting a mammogram and getting a colonoscopy is a mammogram, you find out that you have cancer. In a colonoscopy, you could actually prevent the cancer by getting out this precancerous polyp before it turns into cancer. Just like all cancers that I've discussed that have an inherited component, most are not inherited by Mendelian genes. Only 5 to 10% of colorectal cancer is hereditary. The main players include the mismatch repair genes, which we're going to talk about, associated with the Lynch syndrome. But the first one I want to talk about is fortunately rarer, but much more severe, called familial adenomatous polyposis, or FAP. And FAP is a mutation of the APC gene. It's inherited in autosomal dominant fashion, and it's rare, 1 in 6,000 to 1 in 30,000. It presents with hundreds to thousands of polyps. Like, the polyp burden is so great, you can't actually count how many polyps there are. But essentially, the risk of developing colon cancer is like 100% by age 50. And the management is a total colectomy if an APC gene is identified. Extracolonic manifestations of FAP, including congenital hypertrophy of retinal pigment, or chirpies, supernumerary tooth, although if I worried about everybody with an extra tooth, I'd probably be worried about a lot of people, duodenal and other small bowel adenomas, stomach and fundic gland polyps. Other cancer risks include papillary thyroid, duodenal, gastric, pancreatic, medulloblastoma, and hepatoblastoma. These are rare tumors even in this rare disorder. Here's a family history of FAP. And one other thing I want to mention is some patients have a change in the gene that's not as severe as others, so it's called attenuated FAP, or the APC gene has a mutation, but not as typical as commonly seen. But here we have an FAP pedigree where this father had colon cancer at 47 and died at 49, and you can see the autosomal dominant passion. He had 5 daughters, and 4 of his 5 daughters developed cancer: colorectal cancer at 40, rectal cancer at 38, colorectal cancer at 35, and then an endometrial cancer, and she died at 27. 3 of the grandchildren with colonoscopies were identified to have multiple polyps. So multiple polyps are seen even in younger kids, to the point that in general, I do not recommend genetic testing for children because they can't consent to it, especially if the cancer wouldn't occur till later. However, with FAP, because colon cancer has been described as early as 10, I do recommend genetic testing. A genetic test, any of these children would be at 50% risk of developing this. So if we had the gene in the family, I could do a genetic test, and those that did not have it would not need a colonoscopy. And a cheek swab is less invasive than a colonoscopy. But now we're going to move on to Lynch syndrome, HNPCC, and this is the most common inherited colon cancer. A mutation of 1 of 5 genes is associated, is found in 1 in 400 to 1 in 1,000. So basically 10-fold more common than what I just talked to you about APC. 2 to 4% of all Lynch syndrome, or all colon cancers, are a result of Lynch syndrome. So when I talk about, 5% of patients have inherited, this is mostly Lynch syndrome. In Lynch syndrome, the mutations are the mismatch repair genes: MLH1, MSH2, MSH6, PMS2, and EPCAM. And here's a diagram here of colon cancer. 5 to 6% in the general population. 80% of patients with a Lynch syndrome gene will develop colon cancer. Not that pretty much 100% with APC, but 80%. Endometrial and uterine, 20 to 60%. Stomach, 11 to 19%. Ovarian, 9 to 12%. Urinary tract, and small bowel and bladder. So next we're going to talk about mismatch repair. And mismatch repair is is the problem, what these genes aren't working correctly. So if you remember at the G2 stage of the cell cycle, after all the genetic material has been replicated, we then go through to make sure we haven't made any mistakes. So G is supposed to go with C, not T, C. So in comes the genes, they excise out the wrong base, and they put in the correct base. And this is a small video of mismatch repair.
When DNA is replicated, about 1 in every million nucleotides is incorrect as it is inserted, and this is after the DNA polymerase's proofreading ability is taken into account. But there are enzymes which follow the replication fork, which can then recognize distorted regions where the base pairing is not what it should be, and then they can excise the incorrect bases, and then a DNA polymerase can then insert the correct base, and ligase can seal the nicks. So this occurs along with DNA replication. This is known as mismatch repair. Now, if two nucleotides don't match, clearly one must go and the correct nucleotide be inserted in its place. But how can an enzyme recognize which is the original correct sequence and which is the faulty new sequence? Well, the original sequence has methyl groups on it, and therefore these enzymes look for the methylation of the parent strand in order to determine which is the original correct sequence and which is the novel one which needs to be replaced. In bacteria, a very specific sequence, GATC, is the one which signals this mismatch repair, and it is the adenine in the sequence which is methylated in order to signal the enzymes which is the original parental strand.
Okay, moving on. So tumor staining, and pathology is going to go over this a lot more, but one of the things we now do when we have a colon cancer, any colon cancer, is they stain for the proteins that are made by MLH1, MSH2, MSH6, and PMS2. And if we have, here's a normal control, and here's somebody with a deletion of PMS2, so what we do is we don't see staining for that. So it looks a little bit different. And when there's an absence of staining for these particular proteins, that may mean that they have a germline mutation. They may just have a somatic mutation in the tumor, but when we find this, we then recommend genetic testing for HMPCC. So let's look at this family history, and this is one of Lynch syndrome. And so colorectal cancer at 50, endometrial cancer at 40, pancreatic cancer at 60. That's a rare, but it can be seen with this. Colorectal at 55, endometrial at 30, colorectal at 40. So you might say it's a little bit later than the first pedigree that I showed you, but there's quite a lot of overlap. And for that reason, we actually look at a number of genes. So I probably would not show you a pedigree that's trying to differentiate HMPCC from FAP because they can look a lot of overlap to them. The FAP, it'd just be a far earlier diagnosis. And also, if you see young children with polyps, that would not be normal in Lynch syndrome, but I would not ask you to differentiate between those. So consider genetic testing for HMPCC if there's a family history of colon cancer under 50, or other Lynch-like cancers, uterine, endometrial, gastric, ovarian, a history of greater than 10 polyps, or a colon tumor with the absence of the proteins for MLH1, MSH2, PMS2, or MSH6, especially if there's more than one that are missing. So in summary, in APC, all patients will develop colon cancer by age 50 unless the colon's removed. HNPCC is 10 times more common, and the lifetime risk for developing colon cancer is 80%. And there are other cancers seen with mutations of one of these 5 genes, including endometrial, colon, ovarian, I did not put gastric and small bowel. And that concludes this lecture.